Good day everyone. Today, I'll be discussing one of the most important and widely used techniques in molecular biology, the polymerase chain reaction, or simply PCR. PCR is so powerful that it can change the entire field of genetics, diagnostics, and research. By the end of this presentation, you will understand what PCR is, how it works, and why it is used in almost every laboratory around the world. So, what exactly is PCR? PCR is a laboratory technique used to make millions to billions of copies of a specific DNA segment. Imagine you only have a tiny amount of DNA, too small to study. PCR acts like a photocopy machine for DNA. It mimics the natural DNA replication process, but the difference is we control it, we speed it up, we only copy the exact DNA sequence we want. This is why PCR is extremely useful when the sample is very small, damaged, or limited, like in forensics, medical testing, or research studies. Now, look at the PCR components. First, we have the DNA sample, which is the specific piece of DNA that we want to make copies of. Then we have primers, which are short sequences of DNA that match the start and end of our target DNA. Think of them like a guide rails. Next, we need nucleotides. These are the raw materials or building blocks that will be used to construct the new DNA. Taq polymerase is essential too. It's the enzyme that actually builds the new DNA strands. We also need a mix buffer, which provides the right environment for Taq polymerase to do its job. All these components are combined in a PCR tube, a special tube that can handle temperature changes. Finally, we have the thermal cycler, a machine that precisely controls the temperature during the process, creating the temperature conditions necessary for the process to be efficient. These components work together to amplify our target DNA. I'll explain the specific steps in the next slide, but for now, just remember what each component is and its role in making copies of our DNA. Next, PCR works through a series of temperature changes repeated many times. These cycles allow DNA to separate, bind with primers, and create new copies. Let me explain the principle of PCR. First, we have denaturation at 94 to 98 degrees Celsius. At this high temperature, the double-stranded DNA breaks apart into two single strands. Think of it like unzipping a zipper. Next, annealing. The mixture is then cooled so that the primers can attach to the single-stranded DNA. Primers act like a small starting points that tell the polymerase exactly where to begin copying. And finally, extension at 72 degrees Celsius. This is the temperature where Taq polymerase works best. It extends the DNA from the primers, creating new strands. These three steps repeated for around 25 to 40 cycles, and every cycle doubles the amount of DNA, leading to exponential amplification. This is why PCR can produce billions of copies in just a few hours. PCR is incredibly versatile. It is used across many fields. Let me highlight its applications. So, as you can see, we have DNA cloning, gene expression, diagnostics, genetic testing, forensics, mutation detection, DNA sequencing, drug discovery. We also have environmental monitoring, food safety, research, phylogenetics, mutagenesis, cancer research, and of course, pharmacogenomics. As you can see, PCR is not only for the scientists in the lab. It impacts health, security, environment, and even food industries. Now, let's move on to the importance of PCR. The reason PCR is considered a foundational technique is because it allows us to work with very tiny amounts of DNA. Without PCR, forensics would struggle to solve crimes with minimal evidence. Doctors wouldn't easily detect early infections. Researchers wouldn't be able to study genes efficiently. PCR allows us to copy a single DNA molecule into billions. It gives us enough material to analyze, test, compare, and study, making it essential for modern biology. So, that would be all. Thank you for listening.